We believe we've discovered a unique combination of compounds that have the potential of improving the activity of immune checkpoint inhibitors while also having a good safety profile.
Welcome back, everyone, and thank you for joining us for another Virtual Investor What This Means segment. Today, we are featuring HCW Biologics. My name is Janine Thomas. I am CEO of JTCIR, and I am very pleased to be joined by Dr. Peter Rhode. He is Chief Science Officer and Vice President of Clinical Affairs at HCW Biologics. Welcome back to our platform, Dr. Rhode.
Oh, it's great to be back.
Well, we're so happy to have you. And before we get started, I would like to remind our audience that HCW Biologics is publicly listed on NASDAQ and trades under the ticker HCWB. And during today's discussion, the company will be making forward-looking statements. And I encourage everyone to view the company's latest SEC filings on their website at hcwbiologics.com for the latest information. Okay, so for our audience and for those that are new to the HCW Biologics story, the company is a clinical stage immunotherapeutic company. And our topic for today's segment is the HCW Biologics second generation immune checkpoint inhibitor. And since their introduction to the treatment of cancer in 2001, immune checkpoint inhibitors have been hailed as a breakthrough cancer therapy. And the leader in this category is Keytruda, owned by Merck. So it is on track to generate revenues of $30 billion in 2025. So the big opportunity is that most of the leading compounds, including Keytruda, are facing a patent cliff. And this opens the door for HCW Biologics and its second generation immune checkpoint inhibitor. So now we are going to dive right into what this means for HCW Biologics. Okay, so Dr. Rhode, I'm just going to take a little bit of a step back. How is your immune checkpoint inhibitor an improvement over the existing therapies?
So again, thanks for having me back. I'm delighted to share our recent news on our second generation immune checkpoint inhibitors. So as you mentioned, the development and approval of immune checkpoint inhibitors, which are injectable protein immunotherapies for cancer, have revolutionized the treatment of many kinds of cancers, and they continue to be approved for more indications, combination therapies, and earlier use in the treatment of patients. But the clinical data really has shown that immune checkpoint inhibitors only induce significant tumor shrinkage in less than 20% of cancer patients in their FDA-approved indications. And in most cases, those responses are not durable. So there's certainly a need for improvements in these therapies. In terms of our second generation approach, we've been working in the area of immunotherapies for over 30 years, and this includes approval of the first generation therapies such as interleukin-2, IL-2, and IL-15. And we're still working closely with the clinical investigators that were involved in the approval of these type of products, including immune checkpoint inhibitors. So we have the expertise, technology, methodology, and network to rapidly and cost-effectively test our ideas and then advance the best products into clinical development. We believe we've discovered a unique combination of compounds that have the potential of improving the activity of immune checkpoint inhibitors while also having a good safety profile. We've constructed these product candidates using our proprietary drug discovery and development platform that we call the TRBC platform. And this platform essentially allows us to rationally add other components to the immune checkpoint inhibitors to help them work better. So all of these therapies involve T cells, which are part of our immune system, designed to kill infected cells and cancer cells, and those immune checkpoint proteins that are expressed on the surface of the T cells act to regulate so that those T cells are not overactivated. So it's basically a built-in balancing mechanism. Unfortunately, cancers have exploited this pathway to stop those T cell responses. And immune checkpoint inhibitors, in turn, have been used to reactivate those inactive or exhausted T cells, essentially waking them up again. But there is a lot of evidence to show that the activity of immune checkpoint inhibitors alone is not enough. They lack some critical co-stimulatory activities that are needed for effective for effective T cell responses against the tumor cells. And we've discovered through our preclinical testing a unique combination of immune cell growth factors called cytokines that exhibit the ability to expand these exhausted T cells, or what's called TPEX cells, without causing side effects. And those TPEX cells are located in the lymph nodes that are near the tumor site, and they have been implicated as the major responders in immune checkpoint inhibitor therapy. So these cells have stem cell-like properties, and under the right conditions can change into effector cells, providing those anti-tumor effects. So one of our approaches to improve on the activity of the immune checkpoint inhibitors is to add these cytokine components that will further activate and expand these TPEX cells. So T cell exhaustion is just part of this story. The presence of other immunosuppressive factors in the tumor environment are also another reason checkpoint inhibitors have such low response rate. And we believe we can also improve the efficacy of immune checkpoint inhibitors by equipping them with these other modalities that can neutralize these immunosuppressive factors, particularly one called transforming growth factor beta, or TGF beta. So what we do is we start with the active components of the immune checkpoint inhibitor antibody, pembrolizumab, or as you mentioned, Keytruda, which is the leading FDA-approved checkpoint inhibitor. And then we add these components that can neutralize TGF beta in the tumor microenvironment, and others that simultaneously activate these TPEX cells and promote their infiltration into the solid tumor for more effective tumor cell killing. So together, that's our second generation approach.
Excellent, Dr. Rhode. This all sounds fascinating, and it seems like you're making some progress here in such an important area. So let's talk about the progress that you made. Have you identified a lead product candidate for clinical development?
Yes, our lead product candidate, known as HCW 1140, is a multifunctional fusion protein constructed, again, of those active components of pembrolizumab, along with the cytokines IL-7 and IL-15, and this TGF beta receptor that acts as a trap for the TGF beta immunosuppressive molecule. So in preclinical studies, HCW scientists have recently discovered that IL-7, IL-15, and this TGF beta trap in combination can expand those TPEX cells, as well as to stimulate them into differentiating into anti-cancer effector T cells. And this is all independent of the immune checkpoint inhibitor activity. Thus, so we believe that combining these components with the immune checkpoint inhibitor in the form of HCW 1140 will have synergistic effects to expand this TPEX cell reservoir and promote a large number of them to turn into these anti-cancer effector T cells, potentially resulting in better cancer treatment. So what we've seen in our preclinical efficacy studies is that HCW 1140 can outperform pembrolizumab as monotherapy in immune cell activation, immune cell expansion, infiltration into the tumors, and then immune cell killing of the tumor cells. And in other preclinical studies, we've shown that HCW 1140 is not likely to overstimulate the immune system, resulting in side effects like cytokine storm when it's administered by subcutaneous injection. So based on these results, we're advancing HCW 1140 into clinical product manufacturing and advanced IND-enabling tox studies.
Okay, well, you certainly have made a lot of progress, Dr. Rhode. It's more than just great science or a great idea. Now that you're headed towards your first in human studies and all of the activities leading up to that, what indications have you selected for the clinical development with your lead product candidate, HCW 1140?
So for the initial phase 1 clinical studies, we plan to evaluate HCW 1140 broadly in patients with advanced refractory solid tumors, and then focus on particular cancer types in later phase clinical studies. So these could include patients that have progressed while on the approved immune checkpoint inhibitors, but we're also considering cancer indications such as pancreatic cancer and ovarian cancer, where the immune checkpoint inhibitors don't work well or don't work at all. And in fact, in our preclinical studies, HCW 1140 exhibited potent anti-pancreatic cancer activity and again outperformed pembrolizumab as monotherapy, both in vitro and in humanized mouse models, at dose levels that were well tolerated. So with pancreatic cancer, probably as you know, pancreatic cancer has the highest mortality rate of all major cancers. And in the US, pancreatic cancer is currently the third leading cause of cancer-related deaths, with an estimated over 60,000 Americans diagnosed and more than 50,000 deaths from pancreatic cancer in 2024. So there's definitely an unmet need for better therapies for this disease. And then finally, since immune checkpoint inhibitors have recently been implicated as effective xenomorphic drugs for senescent cell removal to treat a variety of age-related diseases, we're also exploring this opportunity for the future clinical development of HCW 1140. So it has potential to be a broad-based therapy beyond just cancer.
Well, certainly exciting times at HCW Biologics, but I think I'd like to hear from you, and you can share with our audience exactly why. So we've talked about, really important science. We've talked about an exciting development plan and areas of significant unmet need that you're targeting. So Dr. Rhode, in closing, can you describe what does all of this mean for HCWB shareholders?
So I previously discussed our T-cell engager approach, and this is another example of our next generation product candidates that could provide significant clinical benefit to patients and also drive value for our shareholders.
Excellent, Dr. Rhode. Appreciate that. Certainly an exciting time for HCW Biologics and for their shareholders. And this does conclude our What This Means segment featuring HCW Biologic. Dr. Rhode, thanks again for joining us today. So happy to have you back. To our audience, thank you for your time and attention. And to view more events from Virtual Investor, visit virtualinvestorco.com. And if you like what you saw today, I encourage you to visit hcwbiologics.com for more information on the company and to sign up to follow the company to receive their alerts, as well as to follow their social channels to stay current on the latest information. And as a reminder, HCW Biologics trades under the ticker HCWB. Dr. Rhode, thanks again for joining us. Thanks to our audience, and I wish everyone a great rest of your day.